RNA interference was popularized by work in C. elegans, when long double-stranded RNAs were injected into a worm's gonad, a standard way of introducing transgenes into worms. They blocked the expression of endogenous genes in a sequence-specific manner. In eukaryotes, most protein-coding genes are transcribed by RNA polymerase II, which generates pre-mRNAs that are then processed to form mature mRNAs. These mRNAs are then transported from the nucleus to the cytoplasm, where they are translated. RNAi can regulate endogenous gene expression. RNAi can be set in motion by genomically encoded short regulatory RNAs known as microRNAs. In algae, worms, and flies, RNAi can be activated by endogenous transposition. In plants and cultured insect cells, RNAi also has a role in antiviral defense, in which viral double-stranded RNAs are targeted for destruction by the RNAi machinery. When long double-stranded RNAs enter a cell, they are recognized and cleaved by dicer, which is a member of the RNase III family of double-stranded RNA-specific endonucleases. Cleavage by dicer creates short double-stranded RNAs that are characterized by two nucleotide-long 3' overhangs. These are called small interfering, or siRNAs. siRNAs can form a ribonucleoprotein complex called RISC, or RNAi silencing complex. This complex includes slicer, an argonaute protein with an RNase H-like domain called PW. RISC first mediates the unwinding of the siRNA duplex. A single-stranded siRNA that is coupled to RISC then binds to a target mRNA in a sequence-specific manner. The binding mediates target mRNA cleavage by slicer. The site of the cleavage falls in the middle of the region of siRNA complementarity. The cleaved mRNA can be recognized by the cell as being aberrant and then destroyed. This prevents translation from occurring, silencing the expression of the gene from which the mRNA was transcribed. In plants, the aberrant RNA that results from the RISC-mediated cleavage can also serve as a template for RNA-dependent RNA polymerase, or RDRP. This process relies on unprimed RNA synthesis, in which the aberrant RNA is used as a template. The resulting double-stranded RNA is a substrate for dicer activity, which generates more siRNAs. In some organisms with endogenous RNAi mechanisms, for example, fungi, plants, worms, and mammals, RNAi also involves another amplification step. In this step, single-stranded siRNAs not associated with RISC bind to their target mRNAs in a sequence-specific way and serve as a primer for RDRP to polymerize the antisense RNA strand. Such specificity is intrinsically sensitive to natural sequence variation. The double-stranded RNA molecule that is created serves as a substrate for dicer, which cuts it into siRNAs. In turn, these can either unwind and prime RNA-dependent RNA polymerization, or together with RISC, mediate the cleavage of target mRNAs. This amplification, coupled with RNAi spreading between cells, is thought to underline germline transmission of RNAi in worms. RNAi spreading has also been described in plants, but not in mammals.